meaningful, robust data, which is one thing we'll talk about in a little bit. But also it needed to be applied without specialist training or equipment so that managers and community groups and citizen scientist groups could go out and do that. So we had quite a lot of goals for this project. And we were focused, of course, on New York, New Jersey Harbour, where one of the target ecosystem characteristics involves shorelines and shallows habitat and improving the habitat values of those. So our approach, we took a multi-pronged approach to try to develop reasonable protocols for this type of work. First off, we did a literature review to find out what kinds of measurements have been utilised in these types of habitats across the world. Then we did a survey of the sites across the New York, New Jersey Harbour as far as possible to determine the best kind of sites that we could use to test out our protocols. We designed and constructed sampling devices in order to assess communities that are colonising these habitats. We deployed them and retrieved them. At the same time, we were measuring water quality components and doing some fish surveys as well to try to cross-validate potential changes in biological communities with abiotic variables and higher organisms. After that, we did our identifications. We looked at both mobile and sessile invertebrates. We developed a rudimentary fish food index to try to assess habitat quality in that way by utilising the We also went back and redesigned our colonisation device based on some of the lessons we learned in the field. And we've done our data analysis, and that's complete now, and our discussion. So let's go through this in turn. So to start off, we did a comprehensive literature review looking at all studies that have been utilising hard shorelines and sampling hard shorelines in marine and estuarine habitats. And we went from Australia to the Mediterranean to the US, and we tried to incorporate as many studies as we possibly could into that literature review, obviously. Some of the Seattle work is featured heavily in that lit review, so Jim Toft's work and other work from those people has been really informative for our work here. And what we found was that there were a number of common techniques that were often used across these studies. So in the intertidal, usually we would use quadrats or transects, identifying the organisms in the field, or from epibiont scrapes. Whereas in the subtidal, we would often use photoquadrats or basically visual censuses, so for fish surveys along the transects with scuba. Other sorts of ways we can measure do biological community analysis in subtidal and through suction sampling, but also using settlement plates for the sessile organisms. We considered these very carefully, and each of these methods that have been used in all of these studies that we've been looked at were developed for a particular study, for a particular habitat type, but ours needed to be adaptable across a range of habitats and a range of studies, and well as to be used in the future for other non-specialist groups. So we didn't uptake most of these sampling protocols based on either cost. So scuba was not cost effective for a repeatable protocol that needed to be used potentially by non-specialists, by managers, and especially in places that might be dangerous to dive in. And we rejected a lot of these based on practicalities. Although we will get back to the suction sampling and the epibiont scrapes later in the discussion because they have some promise for validation. So some of the problems that we had, or anticipated problems with those past survey methods in this particular location is that both the intertidal and subtidal zones are quite difficult to access on many of these shorelines. And what we were finding, if you look up in the top right there, is that in the intertidal, the diversity and density of organisms was really, really very low in those riprap shoreline areas. So we weren't getting any discernible biota in preliminary analyses of these particular quadrats on the intertidal. So we didn't end up going with that as a survey technique. Subtidal, we trialled surveys with remotely operated cameras to try to get an idea of what was happening on the actual shoreline, to do a survey of what was boated on the shoreline. But these were constrained by really poor visibility at most sites, as you would expect. And this is, I'll just do you a video here. This is from Randall's Island. This is one of the best days, best field days we had across the entire sampling time that we were out there. And we took out our GoPro on a stick, essentially, and we put it down under the shoreline, and this is what we found. So you can see some things there. You can probably see some algae. There's maybe a couple of barnacles or something, but it's not high resolution enough. Not that the camera's not high resolution, but the visual images aren't quality enough to be able to determine community analysis from that. So despite the best conditions possible in the field, this will stop in a minute, it just wasn't going to be practical to use subtidal photo quadrats. We considered scuba surveys, also very expensive, very poor visibility, and not useful for a repeatable protocol. Suction sampler, we thought that it would be difficult to sample a standard area between the crevices, and if we want to get a good idea of community analysis, we need to have a standardised area. Similarly, a scrape is difficult to sample a standard area, and also you end up squishing the animals up. Talking about how to validate those protocols. Okay, so what did we actually do? There's a lot of words on here, but this is just for others to look at when we distribute it. We essentially created a colonisation device that would be utilised in subtidal environments at hard shorelines. So colonisation devices have been used in many aquatic studies, the Hesidendi multiplates, leaf packs in freshwater systems, and also settlement plates on hard substrata for sessile organisms. So what we did was we combined the sort of best attributes of all of these portable colonisation devices into one little neat and tidy unit. So what we ended up having was a steel crab trap where we would enclose insect netting, bunches of insect netting inside, and then a brick on the inside as well for stabilisation, and then some settlement plates on the outside. So the insect netting was to capture detritus and algae moving around the water column, and that would attract mobile invertebrates, the theory was, and they would colonise inside the trap. The settlement plates obviously would capture those sessile biota that are mobile in the water column as larvae. We also included a plastic scouring pad, like a tuffy type thing, to sample the bivalves. That's been a really widely used method over in the west coast of the US and other places to attract bivalves, settling bivalve larvae. And then we had a couple of things that we needed to do. So we needed to stabilise that device and stop it from moving around the shoreline. And so we had these PVC pipes up the back there that would stop it from moving. Okay, so and then we had 5 of these at each site, and we submerged them about 8 weeks, which has been shown to be enough to colonise settlement plates up to about 100% cover. Okay, and so why did we use a colonisation device as opposed to some of these sort of surface sampling? So one of the advantages we were thinking is that it would be utilisable across different kinds of shoreline. So in a riprap shoreline on the left there, it's more structurally complex. We have some animals inside those crevices. They're not necessarily on the surface. And the seawall there, we have potentially lower diversity, and all the animals are present on the surface there. A surface sample would only pick up some of that diversity in the riprap, whereas it would pick up all of the diversity on the seawall. However, our colonisation device would sit down there on the shoreline and hopefully allow movement of animations on PowerPoint. Hopefully allow movement and colonisation of organisms from the crevices into the colonisation device. giving a more complete picture of the communities that are present on in these particular habitats. Okay. However, we'll speak later about how we might be able to validate the comparisons of the communities within the device that we found, as well as the communities on the actual shorelines themselves. So to be really useful, these colonisation devices really need to be a reliable proxy of community structure that's naturally occurring in these areas, and that's part of our next step.
Do you think that the animals are moving into this device, or you're getting new large receptors from the water column into your device?
Mm-hmm.
Are you seeing mature animals or little?
Okay, so the question, just for those remotely from Judy, was is a colonisation device capturing mature organisms or also larvae from the water column? So we were finding both. So of course we were getting larvae of sessile organisms sitting on the settlement plates, which of course that's where they're going to come from, the water column. But we got quite a few organisms in there. We got hundreds to thousands of amphipods, crabs, small fish. We got lots of things inside the actual colonisation devices. So our sense is that we were getting animals that were present on the shorelines themselves that were crawling in, but we were also getting larvae from the water column. So we were hopefully sampling a range of those communities. But as I said, we would need to go back and somebody would go back and double check this and put the device out in the area, and as well as sampling the shoreline itself. We can speak later about it.
We sort of
Yeah.
again talk more about this.
Yeah. There's a lot to talk about here because there's questions about whether or not at the same time, we would really stress that this colonisation device is a very small part of a very large shoreline. And a lot of these animals don't disperse very far. And it would take a lot for a single organism, for example, to move across hundreds of metres of undifferentiated shoreline to just find our colonisation device. So we're reasonably sure that it's a good proxy indicator for the kinds of communities you would get on that shoreline, but that does need to be validated, and we're very aware that it does need to be validated.
But the density of organisms might be
Yes.
off. Is that the?
Well,
Yeah,
I mean, again, the right kind, but potentially.
We're quite sure that the animals are moving from the environment into the trap.
Right.
In addition to settlement from the water column.
Right.
But a significant amount of work would remain to be done to demonstrate that sample in the trap is or is not statistically representative of the environment,
and that the relative community
Right.
abundance of
Right.
the different species within the
Right.
community is the same. I mean, that
Right.
would have to be validated for sure. But we were getting animals. I mean, that's.
Is this only organism-based, or is this vegetative-based as well?
The question from Brian, was it organism-based or like animal-based or vegetation-based? We measured algae as well on the settlement plates, but we didn't quantify that within the mobile traps. So we were focusing mostly on invertebrate communities.
And Brian, the
protocol is for organisms.
the reason for that was actually a little bit dictated by us in the beginning, because there are a lot of established methods to look at living shorelines and wetlands and things like that. What we felt like was missing in this region, and we don't really have a precedent for, is a way to sample really urban shorelines and be able to compare different options at the urban edge. So if you have to, there's been a lot of different techniques proposed, and we don't have a sort of standard way to measure that. And so those are less likely to be vegetated. So while they looked at algae and things like that, they did not look at wetland plants. No, because the shorelines that we're looking at are less planted, but there's, this is filling a hole that we felt like needed to be filled.
so we did measure algae, but we didn't get to the point of being able to actually determine what species of algae it was. It was sort of green or red or... okay, so we designed our device, we had it ready to deploy, and we needed to find some sites, field sites. So what we found in our survey about the different shoreline types in New Jersey Harbour was that they weren't as diverse as we were anticipating. We're anticipating being able to have a range of different levels of complexity across different sites that we could compare. So we examined those aerial maps, we assessed shorelines visually, so and then we went to the actual sites themselves and checked them out and made sure that they were safe and they were easy to access, and we were actually able to sample those. So what we found was we found mostly vertical seawall, lots of concrete vertical seawalls, quite extensive riprap as well, made from boulders, some vertical stone walls at Randall's Island, and some gabion baskets from March's work in Harlem River Park. Lots and lots of piers and pier pilings, which we steered away from. We didn't want to sample subtidal communities that were shaded. We wanted to make sure that the abiotic conditions were, like shading does influence communities substantially, and we wanted to make sure they were all unshaded. So we steered away from the piers, the pier pilings, and we tried to sample these shorelines in clusters, if you like. So we would try to find some vertical seawall concrete shoreline that was close to riprap shoreline and sample them in tandem so that we could compare them, because we know that the water conditions across the harbour and across the harbour estuary will change quite substantially as you move from water body to water body. One thing to note from our initial survey of the shorelines, and that we'll come back to later, is that a lot of these so-called hardened shoreline doesn't actually extend far. Really changed the way we have to think about sampling these habitats. It's not the same as sampling a hard shoreline that goes right down to the benthos, in that we would get a lot of sedimentation down at the lower levels and a lot of sediment scour. And so what we ended up finding was a lot of this sedimentation from boat traffic and from dredging operations and from just water movement would cover a lot of the subtidal habitat, which was something that we weren't expecting.
I'm not sure I understand. So with the riprap edge, for example, it ends up at the bottom.
That would, yeah.
Interface with the river bottom, right?
Yeah, so
The walls.
I mean the walls, sorry Marta. Yeah, so the walls don't actually extend all the way down to the...
They're on a relieving platform and they're just sort of at the side.
It just sort of stops, and it becomes it becomes like just we went down there sampling subtidally, but what we would find is that the wall would stop, and then there would maybe be some wood or something else, but the actual hard seawall that's up the top there just doesn't.
Right.
Yeah, like that, except subtidally. Yep. And so what ends up happening is you get all this this sort of swash going on down near the benthos that we weren't prepared for. We were sampling our colonisation devices designed in a sense for a hardened shoreline that would extend down below the low tide level and stay hardened down the bottom there.
At least
We'll
I'm
talk
sorry,
about that
but
later.
So just one
One
second.
We got to
because
pause, hang on.
We're gonna, go ahead.
Yeah, we have to restart the slide presentation because it's not advancing for those
Oh, sorry.
connecting remotely, not your fault. So let's see what we can do here.
Are you saying that a lot of the shoreline is cantilevered then over the river? Is that the issue?
Yeah. Mhm. Yeah.
There are some that go all the way down.
Yeah. A lot of the ones we were finding just didn't though.
The
So
sites you guys went to ended up either being low-level relieving platforms or high-level relieving
Yeah.
platforms with a concrete base, not traditional gravity structures.
okay. Yeah. Yep. Yeah, I'm not the expert in the structural. I did, but it was definitely something that changed the way that we were thinking about the sampling.
There are plenty of locations.
I don't know where that one
Oh.
is too, actually.
Well, we can go and find them. You just got to know the right people, right? Yeah, we
Next time around.
exactly. exactly.
See, I think it's a map, maybe a map thing.
So gravity well, that's the way.
It's not enough.
I mean, one thing that I know we talked about.
Let's try sharing the other screens. I'm not seeing it.
Sort of shoreline assessment work on your property.
Go back to Adobe. 'Cause I think the first screen is the
One moment, thank you. Some online shopping or something.
technical difficulties. Sorry, guys.
Checking Facebook or something.
Yeah.
And that includes the
It
on-site.
has structure type for the entire city of city-owned structures.
City of, right. And,
I see black screen.
In 7 months, we'll have
Ah,
structure type for the
how did
entire.
you even get to there? I have no idea how that came up.
Do you think?
I don't have Xbox video.
Do you think part of the problem is, are you logged in on Melissa's computer? Is James Lodge too?
So it's one of those stupid Windows 8 computers.
does it have any?
I have no idea how that happened. Hang on, can I just get rid of Xbox?
Oh, yeah. So we were just talking about.
You used to
Yeah, exactly. So I know we had.
I have to
To name the structure type. And then if the structure type could not be identified from the top side, we put divers in to identify whether it's a low-level platform or gravity wall or whatever that structure was. So we did that for the entire, so for all of Manhattan and the 4 boroughs for SBS 10. In 2000, small business services, Department of Small Business.
That's definitely what we need.
Which took over the property from the Department of Ports and Terminals.
Do you want me to get?
for
Is this easier for you, or is it that I should just?
I just used to.
So in 2007,
It shouldn't matter. I
EDC
mean-
went into phase 2 of the
Okay,
mapping.
seems like we're working
All
again.
The remaining city-owned properties, same mapping
I mean, are
criteria.
they're seeing my notes as well, or are they just seeing?
Starting
They are
in
seeing your notes.
December 2014, we started
And the
phase
problem
3 of the
is,
mapping,
I think because
Because we're sharing this.
ORR, Mayor's Office initiative that came out of the SERB report to complete the mapping effort. So again, same protocol for mapping, but we are filling in all the gaps. So we
Never
will
seen
have
that.
100% picture of what the structure type is for every piece of property in the 5 boroughs.
Brian, how's that accessible? Is it available online? The
It's
existing
not,
maps?
it's still being developed, they're in the field.
Sorry.
as we
No, I don't mean
It's-
this,
it's
this
just
died again.
final
No,
version,
no,
but
I-
phases 1
I
and
lost
2,
it.
are they available?
I don't know
Phases
why
1
it's
and
doing
2, yes and no. So, yes, they are available, but they will become even more available. We are, as part of the phase 3 task, we are moving our internal EDC database that houses our mapping data, as well
I guess
as
everyone
well
gets
as
to
our
see my
inspection
notes.
program data. We're moving to a cloud-based system instead of the current EDC-based system, which has firewalls and safety protocols.
Right.
So once we move to the cloud, which will probably be by the end of the year, we'll be able to set up user accounts for people that
it's working.
would like access.
Okay, that's what we're up to.
Have a stake in the information. And that's, part of that is tied to our inspection program. The city just funded a citywide inspection program that we're managing, and we're expanding the EDC's inspection 11 city agencies, and then this database will house all that inspection data as well.
So for those on the connecting remotely, just to repeat what Brian said in case you didn't hear it, so Brian Crane from New York City Economic Development Corporation it has done a mapping exercise involving divers and identifying shoreline type and going in and looking at Manhattan and small business services
Basically
properties.
all of Manhattan, all of the city wrapping up at the end of the year.
And all of the city wrapping up at the end of the year, and that might be relevant to looking at some of this information, and that data may become available to certain interested parties at the end of the year.
Can I say that again? All of the city, even private, will be done by the end of the year?
Yep.
Wow.
Technically,
That
their
would be
contract
very,
goes
very
until the end of fiscal year 16, so June 30th, 2016. But I think that most of the mapping data will be done by the end of the year. The other cool thing that we're doing, which has never been done before, is we're collecting 360-degree photos of the waterfront for the entire 5 boroughs. So it's just like Google Street View. You can click on the photo, it'll come up in the map, and it'll open a viewer window that you can, it's just like Google Street View, but for the waterfront.
That's great.
With high resolution photos. So
Will you be having that as some sort of public visualization tool, or just?
That's all going to be tied through the same database.
Okay.
So we're not opening it to the public, but certainly the folks in this room have a stake in the waterfront. And, we can set up user
That would be
accounts.
amazing.
Excellent.
One
Envision
other
to be a
question.
living document, like if a seawall gets changed into a revetment, for instance, is there a way to update the?
Yep,
you guys
so-
gonna be keeping that up to date?
Yes. It won't be real time, but as part of the theory is, well, yes and no. So for city-owned properties, the theory is that through the inspection program, which will happen on structures every 3 to 5 years, we will capture, if it changes in the first year from a vertical wall to a riprap slope, at worst by year 5, we would know we would be able to capture that data and update the database and mapping accordingly. For private properties, it's going to require new mapping efforts, new funding, new mapping efforts, unless.
Or since DC and Army Corps would ostensibly be permitting all those, I mean, we could probably set up a notification system, I would imagine,
Yeah.
so things can get updated.
Yeah, I don't know if we have staff to be able to handle that, but yeah, I think we can work out. Hopefully, it's not going to change that much. If it's a couple sites a year that transition from vertical to riprap or something like that. Even though the harbor is pretty large, there aren't that many drastic changes that I think we can manage keeping it relatively up to date.
I've made it so people can see more of the actual city.
So we are, yes, in this, as part of previously, no, but as part of this third phase, we're capturing all previously uncaptured assets. So Brooklyn Bridge Park,
We'll definitely be following up. But I want to let Elisa continue.
So, yeah, not my presentation.
We
It's okay.
definitely need a follow-up presentation from Brian Crane on that, but I think we're going to continue. Sorry for the technical difficulty, but that resulted in great conversation, so we'll
Yeah,
keep
hopefully
going.
we're back online for
Okay.
everybody.
Yeah, we should be.
Remotely. Yeah, and that would have been amazing information to have prior to us choosing sites, Brian. Okay, so back to our ideas here. So we wanted to sample shorelines that were quite close to each other, but we wanted to have a seawall site paired, for example, with a riprap site. So we could try to keep those really variable abiotic parameters as tight as possible across the region by pairing riprap with seawall. So what we ended up surveying was we had 6 site clusters across the harbour estuary. We had one at West Harlem Piers. We'll start from number 1. So Harlem River Park, we had a seawall, the gabion baskets, and also riprap. Then we moved over to Randall's Island. We had seawall, concrete seawall, stone seawall, as well as riprap. We had riprap and shoreline and seawall at West Harlem Piers. We had Brooklyn Bridge Park, which was just riprap, but it was a good site to test in a number of ways. We had Ellis Island as seawall, and also Liberty State Park as riprap over in New Jersey, and we had riprap and seawall down in Fort Wadsworth and Staten Island. So I will say now that Fort Wadsworth and Staten Island we lost, and I'll show you some pictures of what actually happened there. So we ended up with 5 site clusters, sampling 5 site clusters. And as I said, we had our, sorry, we'll go back. So we had those site clusters, and we had 5 replicate colonisation devices on each of the different shoreline types within each site cluster. So for Harlem River Park, we had 15 colonisation devices, 5 each on shoreline gabion baskets, and on seawall gabion baskets and riprap, and for all the other ones we had 5 each on seawall and riprap. Okay, and you can see that these shorelines were quite variable anyway, so this was a nice test of whether our colonisation device worked across different shorelines. Through the gabion baskets there at Harlem River Park, West Harlem Piers, riprap, and Randall's Island seawall as examples there. Oh yes, sorry. We kept them. We tried to keep them as equivalent as possible in terms of depth. So we wanted to keep it about 3 feet below the low tide mark at all times. Yeah, to make sure that they weren't exposed during like the low tides. At the same time, we measured standard water quality parameters, so salinity, conductivity, dissolved oxygen. We had a few other water quality parameters that we measured, including nitrates, nitrites, phosphates, and silicates. Chlorophyll A and the suspended solids are pending. Then we also used hobo data loggers to continuously measure temperature and light to see what there might be any differences over time. Oxygen isotopes we measured to determine the sources of the different water that was coming into the sites. And we also did a measure of the relative size of the shoreline substrate. So we did a walk along, we did transects along the shoreline, and we measured the relative amounts of boulders, small small grain substrate. We also did unbaited minnow traps. We just did a single 24-hour sample of that, and we've got really not very many fish. This is almost the entire complement of our fish samples from our unbaited minnow traps across all sites. So 48 minnow traps. We got a few oyster toadfish, as you'd expect, a couple of winter flounders and pipefish, and 1 eel, which was very exciting. We didn't want to, I guess similarly to our device, we didn't want to attract fish necessarily into there. We just wanted to see who was around. Yeah. Yeah, well, the fish was really a side issue, because I know that fish are very attractive of interest, and we wanted to just have a look at what the communities were, but we weren't attempting to actually assess the communities in as thoroughly as we were the invertebrate communities. So we actually got many more fish inside our colonisation devices than we did inside our unbaited minnow traps, as you probably expect. Yeah, of course.
Oxygen isotopes?
Bob can, yes.
So, I mean, we'll see. We haven't measured them yet, but I was trying to get at water sources, and I haven't done this in river versus just to help.
River versus like estuarine marine sources.
Yeah, exactly. Yeah. I mean, groundwater versus recent runoff and stuff like that.
Yeah,
and it may. I mean, we'll see. It's a small piece, but it may not yield anything, and we weren't able to measure them because we are in the middle of a intercalibration exercise. But when that comes down, we'll get them through, and we'll see what we get. Yeah.
Yeah, so most of these water quality analyses were to complement the community analyses. So if we found something really striking or something very bizarre about the community analyses, we would go back and see whether there was anything in the water quality that would point to this particular difference being there. we weren't necessarily advocating for these analyses to be included in a protocol, for example. So we weren't advocating that management groups would go out there and measure. So we went out there and we deployed those 5 devices at each of the replicate shorelines, and then we'd bring them back over 8 weeks. And we did a number of things in order to process the samples and get them ready for analysis. Featured here are some of our harbour school interns, who were really, really great help in the field. So that's Oscar there on the left pulling up one of the devices. You can see it's quite heavily fouled with sessile invertebrates. We would put that into a tub of water straight away and start disassembling the different components. So start taking apart the trap so that we could separate the settlement tiles from the mesh netting. In the field, we would process the mesh netting and pull off any organisms and wash it out. So we got all the mobile organisms out of there. We put Joe there in the middle there is putting them through a 500 micron sieve so that we could sieve out. We would retain all the larger organisms, but we would lose all the sort of planktonic organisms that we weren't likely to be able to identify. We picked out all the debris, for example. Molly on the right there is taking photographs of the settlement tiles, which we would then use in digital analysis. And we would go back. The mobile invertebrates, we passed all of that water through a 500 micron sieve and then preserved them in 70% ethanol and took them back to the lab. The sessile invertebrates, we noted any. Yeah, I know that's a really brief overview of our sampling methods, but this is how we went.
What was your ratio of scientists to young citizen scientists? How much oversight did you?
Okay, we had a whole QA/QC procedure that we did. So we would double check. First of all, we trained all of these. Well, Molly's a scientist too, but the rest of them were harbour school interns. We would have oversight of all of the identifications. We would check them out.
So how many kids did you have?
Well, we had four interns.
Four interns.
And then we had myself, probably 1-to-1. Yep.
But that was,
Or 1 to 2 almost, depending on the site.
I run a high school program up in Piermont every summer, and last year we took one of these devices and put it out for just to see exactly how easy is this work. And we had a college senior who had maybe a year or two of summertime internship, and he was managing a group of 3 or 4 high school students. And the mechanical work of getting this thing properly deployed, and and they were being supervised by one of the Lamont scientists. So it was that kind of a mentor, a somewhat experienced near peer, and 3 to 4 16, 17-year-olds, and they didn't have a hard time. And took it apart and took the stuff out, put it all back together. What they couldn't do at that ratio was the species identification.
That was where we would step in, yeah. But the interns were fantastic in terms of field on-site field management, getting the devices out there. Towards the end, Joe and Oscar and Ava they ran the show. They knew what to do. They would retrieve the device. They'd set up all the tubs, get everything ready, start processing the samples, and with our oversight.
They were used to doing this on the board, though, because they're harbour students.
Well, these kids were.
It doesn't take a long time to train.
Up in Piermont were girls from the Shorelines Leadership School. So they were, hadn't had, the Harbor School's environmental monitoring class or anything like that. They had, you know, they weren't complete newbies, they'd been in the marsh for a couple of weeks, and I think one or two of them had been, the previous summer had spent 6 weeks. So I don't, these weren't just kids who had never been out to the waterfront.
But this
They
is,
weren't
yeah,
trained in this way.
but this is quite, one of our goals for setting up these protocols were to make them so that they were easily done by people who were non-specialists in the field, and this was a really great test for them.
Yeah.
And we've actually been, I'll speak later on about some of the outreach that we've been doing using these sorts of devices and these protocols, and all the different students that we've been able to get involved in these activities, which has been great.
But it's really, the real key there is the species idea.
The species ID was carried out, for the sessile invertebrates was myself, Molly, and Jeff Leventon, with Jeff being the QA/QC oversight guy. Thanks, Jeff. And for the mobile invertebrates, it was a combination of Jeff again, myself, and Molly, who all have experience in those things. And we would supervise the interns. So once we decide on what a particular organism is, they would keep sorting them out, and then we would go back and double check. So we had a quality control plan from the start. Okay, let's keep going. Yeah. Okay, so what did we get? So this is going to be a super overview. We've got a lot of results. There's 169 pages in the report if you feel like reading it. So what in our colonisation devices in the mobile trap, in the mesh netting, we got amphipods, isopods, some crabs, some snails, bivalves, polychaetes. We got quite a lot of diversity in there. Many more fish than in our minnow traps, interestingly. As you can see, over 36 colonisation devices. Instead of about 4 fish, we got about 50 fish. And the scouring pads didn't really add anything to our measures of community diversity. They weren't necessarily attracting bivalves specifically, and they were just generally similar communities to those in the netting. So we advocate for removing those from future colonisation devices. Very briefly, the richness is the number of taxa per device across the shorelines, with the site numbers there and C being control seawall and R being riprap, were pretty similar. We had about between 5 and 10 species-ish across, so identifiable taxa across the different sites.
So the C is what?
It's a seawall, like a concrete seawall.
Seawall or riprap?
Seawall or riprap. So seawall versus riprap, no real differences there. Well, no differences there, and no differences across sites either. Okay, so this is a community analysis, a multi-dimensional scaling ordination of community analysis. Basically, more distance between points represents more difference between the communities. And so we have our seawall control, our riprap. Hopefully you can see the differences there in the symbols, but you probably can't. Gabion baskets at Harlem River, and also Stonewall. And what we really saw, that the locations clustered out rather than the shorelines. So communities were most similar within locations, and the biggest differences were across locations, between locations. So the site numbers there, all the communities at the different sites were more similar to each other than between shorelines. So for example, the riprap communities weren't more similar to other riprap sites.
The built environment complexity differentials. You couldn't really even see the differences between surfaces for the different, in comparison to the differences between places.
The differences between locations far outweigh any differences between shoreline types.
But the variable is what?
Whole community of abundance and diversity. It's a multivariate analysis.
So is the take-home message, we should just forget about doing these?
Not yet. Not yet.
Can we maybe
Take-home messages. We haven't finished.
time-wise, we have
I know,
about
there's a
half
lot to
an hour
go.
left, so I think we should just hold all our questions and then we can go refer back to the slides until the end, and then we can get into discussion. So let
No, the
Elisa finish.
take-home message is not there yet. We'll get there. Okay. So, Okay, so what did we get in the sessile communities? So we got a range of organisms, as you'd expect, solitary and colonial tunicates, so Mulgula manhattanensis, Botuloides, Botrytis species, a lot of tube-dwelling amphipods and polychaetes, bryozoans, macroalgae, of course. And what we did there is we digitally analysed those photographs. We analysed the digital photographs with 100 points. We debated whether to do a grid or stratified random, but we did stratified random in the end. So we'd split up the plate into four quadrants, and then randomly assign 25 points to each quadrant. Okay. So we had plans to do statistical analysis of differences in percent cover, taxonomic richness and evenness, and as well as community structure. We tested 4 different materials. So we tested acrylic, ceramic, slate, so a stone proxy, and also wood. And these are just examples from Liberty State Park of the differences in communities. But what we ended up, the broad picture was that we lost replicates at all of our sites. So we ended up having so much replicate loss through a variety of reasons, and I'll tell you in a minute. And there is quite a lot to go, so I'll try to be really quick. So we weren't able to do our univariate analyses across materials and across sites. We ended up analysing the different materials separately. We were able to do those community level ordinations though, and most replicates were retained at Liberty State Park. That was the best site for keeping all of these colonisation devices intact. Very broadly, there were we do need further work on that. Okay, now I've just highlighted the things that you should look at here. So these are just very broadly on the acrylic plates, taxonomic richness across all the different sites, and the sort of differences between the algae cover and the invertebrate cover, very broadly. Down at Liberty State Park, as I said, where we had all of our replicates retained, and say we had the highest diversity, highest taxonomic richness at about 4 or 5 taxa per plate, and then the seawalls had mostly invertebrates, and then the riprap was reversed with mostly algae, and that was something that we expected in that we have higher light penetration on the riprap shoreline and higher algae as a result. So I'm just giving you super broad results because there's a lot of them. So this is an example analysis example from the acrylic plates. So commonly we would get lots of algae, and we'd get lots of algae and sediment detritus, and maybe some tube-building amphipods or polychaetes as well. Less commonly we will get colonial tunicates and solitary tunicates as well. So this is the kind of mix that we would find on our sediment plates. And you can see there's varying amounts of each particular taxa across plates. But as I said, we lost so many replicates, it was difficult to compare across materials and across sites. We got some. We didn't get as many as we were expecting, but we did get some, and I think it might have been partly length of deployment as well. We're starting to get them more towards the end, and we were doing it within summer, and the bryozoans tend to settle a little bit in spring, a little bit in cooler weather. So this is an example community analysis for the acrylic plates. Again, we were finding that the communities were generally more similar within locations. So
sorry, we're gonna go. We lost it again. We're gonna have to go in and out. So. Sorry.
That's all right. I knew I had it. It's too long.
Yeah, sorry
okay.
Everyone on the webinar.
Well, just a quick question, and what do you mean by replicate loss? Like you physically lost
Yep.
the
Yep.
plate?
Yep.
So, I mean,
Plates would fall off, or the whole device got smashed.
we really
I'll
did
show you.
redesigned it somewhat, sort of shifted the profile a little bit and changed the materials. And then the second version was much more successful in terms of stability and stuff. But, I mean, it was a protocol development project, so, that's what you learn in the
Yeah,
process.
yeah, we definitely learnt
Yeah.
from that.
Actually, I'm pretty sure
That
Brooklyn
makes it
Bridge is
easier.
being repeated.
Because it's really
We
not,
didn't
I mean,
try to systematically
we really
do
don't
that.
have
It
much
was hard
time to
enough
do
to
the
find sites
animations.
that were accessible to the middle schools where the teachers that we recruited happened to work.
All right,
So we're gonna try this again. If we keep having
keep-
problems, we might just have to, kind of,
chat.
yeah, exactly. So we'll.
Okay, I'll try to run through this fast as I can because there are messages. Okay, so these are the sorts of results that we were seeing. But okay, some of these disadvantages of our original colonisation device and some of the motivations for us to redesign the device and some of the lessons learned from this particular protocol development are in front of us here. So the device works really well on those near vertical hardened shorelines that go below low tide level, which is what it was designed for. It works great on those. So it worked really well at Ellis Island, for example, where that seawall goes right, like a lot further down than it did at some of those other hard shorelines. The cage itself and the attachment of our sediment plates, which was a difficult thing to try to figure out, weren't always strong enough for the conditions. So on the right there, we have a cage at Brooklyn Bridge Park, which got crushed. This was when Hurricane Arthur was coming up the coast. And some of the plates got lost. So we lost a lot of plates, we lost a lot of traps. We had storms blew our devices up onto the riprap shoreline, and so they were exposed for a period of time. That was the ones at Staten Island. Okay, so, however, we did get, so that was one problem. It wasn't strong enough. We lost our settlement plates. So that's why the loss of, it wasn't heavy enough to withstand these storm conditions. Another disadvantage or something that we found was that a lot of the those shorelines that didn't extend right down to the benthos we would get a lot of fine sediment. We would get a lot of sediment scour down towards the bottom. So we didn't get a lot of sediment on the outside of plates because we had that sediment scour, but we did get a lot of diversity on the inside of our plates, the ones that the surfaces we weren't particularly interested in. It was least effective on those low gradient shorelines, as I said, so they would get blown up the shore, or on those areas where the hardened shoreline didn't extend to the bottom. All right. And so really, like, we weren't really testing what we thought we were testing, which was that we were looking at the hard shoreline habitat, because we would have this hard shoreline, for example, up at high tide, and that was fine. We'd have our device in there, but at low tide, the device would be sitting down there near the bottom, and we would get a lot of that sediment movement and sediment scour coming in, restricting settlement on the settlement plates. And that is exacerbated by things like this, which would happen in Harlem River, the circle line going round and round. When Kate came out with this one day, and we would see the sediment plume coming out from the end of that boat. So we needed to think of a different way of doing things, just because it's fun to see. Like, so Harlem River Parks, that's some of this fine sediment would really coat our device at those places. You can see that there. And this was a test that we did deliberately to show that this device would not work at high energy shorelines. So this is not an actual sampling device. It was a setup essentially. So this is not the roughest, but right, as I said, that's a setup. we realised that that was not good. So what we did was we redesigned the invertebrate colonisation device. So this is something to take away after this presentation. We were inspired, in a sense, by the robust oyster garden cages that we were using out at Harbour School. And so what we did was we used that really strong steel, vinyl-coated steel mesh netting to construct a new device. And it was lower profile, it was less likely to be blown around. We had 4 bricks instead of 1, so it was much heavier. But essentially, we kept the components that were working. We kept the plastic netting and also the settlement plates. You can see in that side view there, we would have this. We also had a backing panel to kind of protect from any scraping of hard shorelines, and also for stabilisation, and also to separate those settlement plates from the hard shoreline, and also from any sediment scour. We had settlement plates at different orientations, right, so that we could measure the effective orientation too. And the whole thing is much lower in profile, so it doesn't stick out as far from the shoreline. It's more streamlined.
And you don't use those PVC poles anymore?
We don't need the PVC poles at all on that because the whole thing is heavier. It has a better sort of centre of gravity, if you like, and it doesn't flip around because it's a lot shorter. We don't have one here, but unfortunately. I could jump over there to Governor's Island and get one for you. Okay, so there's some pictures of it.
The opening size of your grill or fencing?
Oh, it's 1 inch by 1 inch by 1 inch.
So you're excluding anything larger than 1 inch?
Looks like it, yeah. Still getting fish in there.
Still get fish in there.
Still get fish.
It's
But
a
we're
fish-
not as interested in fish. It's not a fish colonisation device.
It's not a fish trap. Right, we weren't trying to build a fish trap here.
No.
But it is, with the little fish, the minnows still do, get in there.
So here's a few sort of pictures of this thing. It's a little hard to see, but it's much more low profile. If each of those is 1 inch mesh, it's only about 4 inches high, and it extends for about a foot. So it's low profile, very heavy with this brick. It's very sturdy, and that is a device that's been pulled out of Brooklyn Bridge Park. It had been sitting there for 8 weeks, and it's intact. We haven't lost a single thing. The other thing that we did to keep those settlement plates on is that we have them in this prism arrangement so that we have different orientations. Then we make a cover based on that actual wire mesh as well, and we would secure that cover over the top of the settlement plates, basically to hold them on, rather than using cable ties or something like that.
And if you have, 2 or 3 hours, in 2 or 3 hours, a middle school, high school, middle school science teacher can build one of
Yep.
these in 2 or 3 hours if you have the materials there.
Yep, and it's relatively cheap materials. We went with a ceramic settlement plate as a standardised unit across all of our traps. They're easy to get. Just go to Home Depot or something like that. Get an unglazed ceramic plate, ceramic tile. So it's a house tile. You don't have to go to a specialist hardware store. And ceramic is utilised widely across these sorts of habitats to kind of assess invertebrate communities, and it provided a pretty good proxy. So that's.
The picture on the left, is that
Needs. So this is just a diagram. Obviously, this is the invertebrate colonisation device and how it would lie on the different types of shorelines. So instead of being only applicable to something that was a hard shoreline that would extend down to the benthos, which was our initial design, this one is adaptable to different kinds of shorelines. So with that backing panel as well, it would stop any scour from small boulders down near the benthos. And the settlement plates at different orientations would hopefully mitigate some of that sand scour down near the benthos as well. So on a riprap and a hard shoreline, this is how the thing lies. So this is the sorts of time involved that you would need to use our proposed protocol using those redesigned colonisation devices. So construction, it's relatively cheap, as you can see. probably an hour if you're really used to it, or maybe a couple of hours if you've got a couple of
$50 an
students.
hour, I got a kid that could make a lot of money.
And we pay
Materials.
materials. Sorry, that's material costs. Okay. So, and this is probably a conservative measure too, depending on how many you're making. This is, you can buy this wire mesh in large rolls, and the ceramic tiles are pretty cheap. You get about, for deployment and retrieval and collecting these things, you might want to take about 12 hours, so 4 hours actual on the field time, and then 12 person hours. Identification of mobile invertebrates, we were monitoring as we went how long the students were taking to identify all of these mobiles and how long the extra checks were taking, and it averaged out about 2 hours per sample. So per site, if you've only got 5 devices at a site, it will take you about 10 hours to sort this out. The identification of sessile invertebrates across the specialists we had, myself, Molly and Jeff, we were taking around an average of about an hour a sample. This is for 100 points. So I would expect that to increase with non-specialists, but we can also modify the protocols to suit. And identifications from the scouring pads, as I said, we wouldn't recommend that being in the final protocol, but that took about half an hour. It was pretty easy to do. So for a full site, you're looking at about 50 hours of person-hours to construct, deploy, retrieve, and ID everything and do all the analysis. All right, so what are we up to? So I'm mindful of the time here, so I'm just going to keep on going. We'll have the questions at the end. Okay, so so we developed this protocol, and some of the side benefits is that we've managed to get this device out to lots of different community and education groups around the city. So currently the colonisation device with modifications is being used by the Billion Oyster Project, and it's been called the Restoration Station. And at the bottom there you can see some Harbour School students with a restoration station that they've built. What they've done to modify it is to essentially stick an oyster cage on top there. So we can monitor oysters. What we're doing is monitoring oyster growth, as well as the mobile invertebrate colonisation, as well as the sessile invertebrate colonisation. And I'm currently involved in modifying those protocols and adapting them to school groups, and involved in the teacher training as well.
And Molly Thurman, who was one of the doobies on this project,
she's now at BioBus as an educator, and she's using that with her school groups out there
she's
too.
developed a whole after-school curriculum for shoreline monitoring based on the device.
Yeah, and so up the top is Latasha Wright from BioBus using a modified colonisation device for some of those students too. And as Bob mentioned, he's used it last summer at his field science summer school. So it's really, it's getting out there. Also, it's just been provided a really great opportunity for collaborating with interested folks like yourselves and with all of these different organisations. We had those high school interns from the Harbour School, gave them some excellent experience in this research project, which is really current and really happening within their region. Also, they were fantastic in providing input on how the methods were going, because they were all diving majors. They all knew about the subtidal and intertidal environments within the harbour, and they could advise us on the design of our device and some of these sorts of questions. And I really think that the collaboration that we've made here has provided additional momentum to trying to adapt these protocols and to try to think about shoreline restoration research. So some of the next steps, and we would advocate for the priorities for the next steps would be to cross-validate this colonisation device as a method of assessment. So essentially, are the communities that exist on these shorelines equivalent to the ones that we find in the device in terms of both their abundance, their diversity, and their relative abundances as well? So additional measures we could use, and we might want to use photoquadrats, but we would start trying to maybe trial them out in less turbid areas than the New York Harbour. Epibiont scrapes definitely show promise, and we might want to use other artificial substrata. We might want to, for example, take out some of the components that show real promise for enabling or facilitating colonisation of invertebrates, such as the mesh netting, and maybe put it in the actual shoreline. So, going to open up to the floor about ideas about how to cross-validate this, but also try to be mindful of the limitations of these sorts of cross-validation techniques. So, the relative importance of those quantitative data and qualitative data. So, Epibiont scrape might get you a lot of information about biomass, but it might not necessarily tell you a lot about species analyses, but we can talk more about this. We need to think about the precision of our results. We need to think about the accessibility of these different shorelines too, and can we actually do some of these analyses? All right. So other considerations for our validation are to think about the length of deployment of particular colonisation devices. So we also tested 4 weeks versus 8 weeks deployment, and we were getting very different assemblages. So if we wanted to cross-validate, have to make sure they're done the same season across the same amount of time.
The 4-week, 8-week, was that overlapping? I mean, was it?
Yes.
Okay.
Yeah. So the 4 week was basically taken out early, and the 8 weeks was taken out later.
But you didn't put another 4-week back.
No. It was sort of time limited logistics.
But that's the kind of thing we should.
So successional processes
is
is something that we need to think about. And we need to be very mindful of what sorts of questions we're asking and make sure that our methods fit the questions.
The 4-week and the 8-week, it's not the same stuff, just proportionately more for 8 weeks.
No, it's a different. After the 4 weeks,
Right.
it's after the 8 weeks.
I mean, if you remember back when we first started, we talked about, that there would be a succession, there's a natural succession, and that, and the whole project was only a few months, right? So that, ideally, if you wanted to do this kind of monitoring, you'd have this device in for several years. And
Yeah.
Just to see if that plate system
Right.
is actually useful.
Right, but I mean, the shoreline you're testing has been there for years, right? So and you would expect that in the first few months, certainly in the first year, as you go through the seasonal cycle, you're going to see a shift in community assemblage, and it's not until at least a year after you put this thing in the water that you'd have a shot at.
It depends.
Okay, but it was good. You have to go
The
through.
time scales of putting these things in the water need to kind of match up with the time scales of the disturbance regimes along those shorelines.
Okay.
So it's depending on how many storms have come through and cleared up space on that particular shoreline. You might get assemblages on the shoreline might be at different successional stages than the ones on your colonisation device.
Jeff
so
Levinson
that's something to be mindful of. Jeff will have something to say.
Suggested that one part of succession is also the displacement of settled sediment by sessile organisms. So
there's a lot of So consider that. Yeah. Okay. And also consider the measurement of those other parameters and how close they are to the colonisation device and how likely they are to sort of vary spatially. Seasonality of validation and the location of cross-validation. Obviously, we need to find accessible sites that are measurable, and also we need to think about the recruitment windows. What I'm saying is that there might be seasonal differences in the recruitment of different species across different sites, and so more information on those sorts of recruitment patterns need to be considered. All right, Brian.
So the plates.
Yeah.
What's the point of the plate? Is it supposed to replicate? Because the materials that you guys use, wood. There's certainly wood in the river, but there really isn't ceramic or acrylic. Why wouldn't you use plates that replicate like a concrete plate, and timber makes sense, but we're not putting that much timber in the river anymore. Or why would you use a steel plate, or why would you use like a calcium-rich something that to develop muscles or moisture to see? I just don't get the material type selection.
Yeah, so the question for the remote people was why did we use a range of materials and why did we use those particular materials? The materials question is one that can take a long time to discuss, but the materials question, we wanted to use something that was easy to obtain by different groups. We trialed things like concrete. We wanted to something that's easy to obtain by different groups, was able to fit on top of our device, and provided a standardized area for us to analyze digitally.
So the thought is these are close enough or similar enough?
It was a proxy, yeah, and some of Jeff's work has shown that the ceramic, actually you end up attracting communities that are reasonably similar to that which we would find on a concrete seawall, for example. So there are many questions we could answer about these things. Creating tiles out of concrete, for example, we trialed doing that. That was something that fell through. Little steel tiles.
But that would be an
Or a
interesting
possibility.
experiment, right? To put some out with with ceramic and make up some concrete plates, which is not quick and easy, but you can do it and put them out
and the time
preparedly.
scale.
But in the time we had to
Time we
do
had,
this,
we
that
just-
was the
yeah.
experiment.
But it's an ongoing question, and what we initially wanted to do was we needed enough replication of a particular material to analyse these communities in a robust manner. Obviously, with our loss of replicates and with our material differences, we didn't end up with that. So in a validation, I would advocate for a single material across all replicate devices that could then be analysed. The communities could then be analysed between shoreline types and between locations in a more comprehensive way than we were able to do.
Yeah, I would think that for revalidation or validation, I think it would make sense. I guess it depends on what the end use of the protocol is. If the design of the protocol is to determine the best construction materials for shoreline construction type to promote this type of organism benefit or growth, then I would guess you would want plates that would replicate or signify the types of materials, but then also common construction material types that you would potentially would put in. So
Yeah.
maybe like a fiberglass or
I definitely agree.
like that.
But our goals here were not to figure out what should we construct shorelines of in order to get the best habitat. It was to try to design a way to measure community.
I'm just thinking the
Yeah,
next step.
exactly. Next
Absolutely.
steps, absolutely.
That-
We'll go forward.
That was discussed. I think one of the things that becomes challenging is that it's then for this, it would have been sort of an experiment with an experiment because you're losing control when you introduce more types. And so they try to do some of that, but
Yeah, it was a compromise between getting as much data as we possibly could and also doing these materials assessment. But it's definitely something that we can consider
wrap it
for the
up and
future.
then
Okay,
have a
so
short discussion.
the summary of our protocol development is that it's a really promising technique to assess habitat quality in terms of the diversity and abundance of biological communities. As I said, we want to cross-check what we've been getting with our invertebrate data with those abiotic data and also cross-validate across current shorelines. It's suitable for wide uptake across regions. The materials are easy to find and they're pretty cheap. Suitable for non-specialists. You don't have to get in the water. You don't have to have boats. You don't have to have any of those expensive equipment. And we're really working hard to make these protocols as straightforward and as easy to use as possible for multiple users across the region, including school groups and citizen scientists. Bear with me, a couple of extra slides. Okay, so some of these things, with all this interest in living shorelines and eco-engineering, these sorts of protocols and monitoring and involvement of community groups is really taking off across the world, as you know. So here's a potential further partnerships. I'm involved in the Billion Oyster Project. I'm also involved in the World Harbour Project in Sydney, and I'll give you a little bit of a detail on that in a minute. The Nature Conservancy schools. Write me an email, right? I'm at shoreconsulting@gmail.com, and the website's going to be live soon. So for interested partners in partnering up in eco-engineering, the World Harbour project in Sydney is looking for New York partners for its green engineering project, with the aim to run a global experiment looking at techniques on how to reconstruct seawalls to encourage restoration of bivalve communities. So Sydney is one of the places where a lot of work has been done on this on seawall construction and the benefits of hard shorelines for different invertebrate communities through Tony Underwood and G. Chapman's work mostly, and that whole lab. So what these guys are trying to do is to test a range of methodologies for enhancing ecological enhancements of hard shorelines. So with their hypotheses, of course, being that we're going to have adding complexity will increase community diversity and productivity, and the effects are going to be more pronounced at those lower latitudes and higher latitudes. And in the map down there, you have a map of the current World Harbour project partners across the world. And the benefit, this is interesting hypothesis, and it might speak to some of the lack of differences that we found in our particular protocol development, is that those benefits might be more limited in these highly polluted harbours. So contact people at the World Harbour Project if you're interested. They're looking for New York partners now. They're really keen for people to go out and do this sorts of work.
We had some initial conversations with some of the
Yes, yeah, it's great. So, and I'm in touch with them regularly, so you can always talk to me as well. So thank you, and here's a list of all the people that helped, and we can open up for discussion. And thanks for bearing with what was a rather long presentation.
So I first want to say thank you to the whole team that's online and in person here for taking on a really, really challenging project. And I don't want to, we don't take that lightly. We know it's a very hard thing to do. And I think this takes us a step closer to really getting something where we can say something about it, about our shorelines. For those of you that have to go, it's 12 o'clock. We understand that. So I think, if you have to go now, we can take your questions following, and I'll try to follow up with you and get them answered. For those that might be able to stay for a little bit, I think Elisa has at least a little bit more time, so maybe we get a little more discussion. I think for us, we're really interested in hearing from you guys. I think the thing that we're sort of challenged with right now is through this project, the team has been able to demonstrate at least clustering of differences between sort of local effect successfully. I think, what we're trying to figure out is how do we take a next step towards making sure we can test differences between shoreline type, if that's possible. I think we have to recognize that it's a big challenge. It may not be easy to separate that regional effect versus local effect. So the next step that I see after looking at all of this is really focusing on that validation piece, which Marcia brought up and Judy and others, to see if it's possible. And I think it's resulted in at least a great step forward in that direction, also a great step towards using something that's for schools. Yeah, for that.
Yeah, that's right. I mean, we can use some of the devices that are out there currently and that are going to be out there for the next 2 to 3 years under this project with the Billion Oyster project to kind of cross-validate.
Yeah. So I'll bring up one question from Kristen Marcel. She asks, do you have ideas for how to address the challenge of identifying relative ecological benefits of New York City hardened shorelines, distinguished between valley riprap or seawall? I think that that gets to the question we were just talking about, but maybe you can. So she's basically asking about differences between
shorelines. How can we?
Yeah.
Well, I think that, as I said, I think the first step is to make sure this technique is representative of the different shorelines, and then just have replicates there. I mean, I don't think that you necessarily have to do any anything too different to actually find a difference. And if there is a difference, is a clear difference between those shorelines, we're going to find them through this technique because we found clear differences between locations. The technique is robust enough statistically and to find those differences between locations. So if those differences between shorelines exist as well, we'll find them with
So
this sort of sample.
That validation would involve epibionts, scrapes,
Yeah,
and suction.
potentially.
And that kind of stuff.
Yep.
I think the first thing would be to do is validation, and because otherwise you don't know what, we simply haven't had time to validate and know that the technique is representative. If it proves out, then the next thing is to get better numbers. I mean, we need, if you want to compare, I mean, this would indicate that if you want to compare riprap and seawall in a fairly, small spatial environment, you're going to need more replicates in order for those statistics to fall out. And
Yeah.
it is simply, I mean, you were talking, Kate, about the local versus regional effects. It may actually be that if you put riprap and seawall, interspersed near each other, that the differences are not strong. It may be that you require a certain spatial scale in order to get the benefits, or what we presume to be the benefits of a more structured, more complex shoreline.
Yeah.
But you need numbers. I mean,
Well, yeah, we had enough numbers to separate by location.
right,
But
that's a
we
stronger
acknowledge
effect. You're not going to
it's
get
a strong effect. Yeah, some of these more subtle effects we're going to need. We're going to need higher replication, and we agree with that.
One thing that I would take out of that is it might be very useful to know that in certain locations, it would be better to do interventions like this than in other locations. Some locations
That's true.
Mhm.
Yep.
seem to lend themselves to doing more naturalistic shorelines if possible than other locations. That's a really
Yeah,
big
that's
take-home
true.
message for the city. If we can do a few of these in some places.
Where should we
but
focus
not
our
everywhere,
effort?
Right.
where would it make the most difference? That's really useful.
That will be a very useful outcome, I think, if we do more of these.
Ask something kind of similar along, I mean, like, do you have way? In other words, you guys found that your shorelines were kind of similar, right? Same locations, and if we know the different shoreline types that are out there, what's the level of saturation for these devices?
I think
Well, that's why, I would
It's
totally
the water
agree.
quality,
It is
okay.
the water.
But and also
So-
the water movement too. If it's a longshore
Yeah, I
water
mean, it's
movement,
exactly.
like steady alongshore water movement, you're going to get larval delivery across that length of shoreline.
What are we
But
talking
if
about,
I
like
may,
hundreds of sites?
no, look, if I may say a bit more, I think our level of planned replication was sufficient. It's just we lost replicates.
Yeah.
And with this new device, we've had 0 losses of any replicates across our test site, which was Brooklyn Bridge Park, which sustained quite a few storms in the test region. And we would think that the 5 replicates per shoreline type, per 100-metre shoreline, would be sufficient for an extra thing. And I would also I'd also hesitate, just to be careful, if just because we want there to be differences between these shoreline types, it doesn't mean there are differences. And more looking isn't going to create a difference when there are none. So we can potentially use things like power analyses to be more confident in our results. And that I would advocate for using something like that in a statistical analysis of repeated sampling. But yeah, don't try to make something exist when it's not there.
It's great for vertical seawalls and not so great for
The original device, yeah, the original device isn't great.
Intuitively, you want to think of shorelines that are more similar to a natural shoreline. The more similar to a natural shoreline, a slope that you get, the less effective this particular method.
The original one is. The
No,
second
but the
device, the
second
redesigned
device worked
device is fine. It's great on both.
On both.
It really is. And
We put it out on Brooklyn Bridge Park, where you have the sloped riprap, and it
Yep.
survived very well. I mean, got nice results from it.
Yeah, and you still get colonisation, community colonisation of these things.
Two points. One is that it appeared that the improvement of riprap over seawall was very minor. Which was kind of disappointing. Hoping that riprap would be
I mean,
a lot better. It appears that it wasn't. But obviously it needs to be, the whole thing needs to be validated.
Right.
And the validation that needs to be done is looking at what is actually there.
Right.
Yep.
Rather than more of these
I think-
devices in more places, you've got to find out
Right.
what is actually living on these places.
That's what we said. Yeah, we have to go out and deploy the devices in a place where you're also going to do scrapes or suction. Yeah, and
So I
compare them.
think we understand that that's a really important next step, and particularly because there have been studies published. Dave Strayer's published a study looking at riprap versus other types of urban shorelines, and found that it might be better than a vertical seawall, and looked at some of that.
Seawall with flower pots on it.
Yeah, exactly.
definitely, yeah. I mean, and we also might hope that riprap is different, but it may well be that it isn't.
This is great. I'm an architect coming to this.
Yeah.
That's
good.
But because we're very interested in the way the formation of the seawall and the way in which the water and land meet and the way people interact with the wildlife and so forth. One of the things that is a question for me here is the scale at which you think. I mean, you say there's either riprap or a seawall. Yeah, what it seems to matter is the flow of water. Right?
Yep. Seems like.
I'm not a biologist, right? But the directionality, the incoming, the flow, and so forth. So is the water, is the scale of that difference? And is it on a sort of leeward side or windward side? Is it upstream or downstream? How do you begin to account for things like that? Because you can design something that is different on the upstream side than the downstream, or it can vary in different tidal conditions. There's a much finer grained design that we hope to be able to bring here to the edge. So other ways you can begin to feed
I think that's good
more
thinking.
about a finer grain by thinking of a larger grain, right? Because the behavior of the wildlife at the edge is not, I think, purely on.
So
Right.
I'll just, can I just read?
What I would-
I better repeat. Can I repeat a summary? I'll try to summarize the question for the remote.
Thank you.
So the question was, how do we think about the scale of water movement and tidal influences when redesigning shorelines?
Right.
Essentially,
is the
very,
scale
very
more
briefly.
The scale of the texture? Or the
So,
scale
right.
of the
So,
quality?
Right.
So I think
Yeah.
that's right.
Yep.
So I think the short answer is that we don't know what the
It's an
scale
interest.
is,
Yeah,
right?
it's
Right.
So that if we're able to validate
Exactly.
these results, so we go out and we put these things in, and we do
resounding
scrapes,
yes.
and it turns out that the device is representative of the environment at this aspect. And we do, another season, and it's all fine, and this checks out. Then I think this indicates that
Trying, yeah.
the fine scale is not what's determining, right? That there is something I like the way you're thinking, that there's something about the shape that's impacting the water flow, or maybe it's the water chemistry differentials. We know there's a lot of differences, and a beginning, I think, is that we see significant differences in place. And you could start from that scale of data and work your way down to a finer scale. So this is like, this is the 10 to 20 kilometer scale, and you could say, well, what does it look like at a kilometer? And actually, based on what's the differences that we can observe, try to see which types of shape of shoreline are more diverse and more productive, and which ones are getting. And I would just think I would do it empirically. I mean,
And that's what the World Harbour Project is trying to do. They're trying to do experiments across harbour shorelines worldwide to look at the differences in complexity of hard shorelines and how that attracts diversity. But I think that conversation about how about the shape of the shoreline itself is being lost even in that particular conversation, and that they're looking more at the scale of what the organism experiences, or the fine scale on the actual shoreline, rather than upstream, downstream effects or intertidal effects. So an architect's perspective on those sorts of experiments would be really, really interesting.
If we can identify within the harbor the locations that are working better in terms of diversity, then we can attack how much of that is water chemistry. That's actually fairly straightforward. And if it's not water chemistry and it really is shape, then you're at the beginning of an interesting, process of defining what that is.
So
Those two things related, water chemistry and shape.
Yeah, they are, but not, but here it's also, do you happen to be next to one of the 14 treatment plants or not?
Yeah, a little bit of input from Jeff is that larval supply and distribution around the harbour in terms of hydrodynamics is going to be really critical to trying to designing devices that are actually going to harness this diversity. Dennis. So Dennis asked, were they put out on flat beaches? And the answer is no. Essentially, we were looking for near vertical hardened shorelines and trying to compare those with lower profile riprap. So no, we didn't put anything out on soft substrate intentionally, and we didn't put anything out on those super low profile shorelines. It was the project was, the RFP really called for trying to assess hard shorelines.
Again, so to Brian's point earlier, I think that the idea was that there are a lot of existing protocols, maybe, and there are for beaches too. I've actually conducted some of those. I think that the idea is that there's a lot of existing protocols for things like trying to get that sort of fine signal between the range of really urban shoreline types so that we can look at new.
Yeah, I was thinking more that in the validation where you've got in your beach environment, it's much easier to sample. And when you do your validation, you get at that question whether you're actually creating new habitat or you're actually sampling something.
Yeah.
And we do know a little bit about, that's where we see the largest differences among habitats and fishes, juvenile fishes. Anyway, there appear to be lots of juvenile fishes in these beach shallow water areas. And so I would just possibly make that recommendation that, right, for your validation, you're going to want to see areas where you've got the greatest differences in abundances and diversity. So I want to think about that.
Yeah. So the comment for remote people was that beaches and other types of habitats might be a good place to start for validation of the technique. And I would definitely think that for validation, you'd want to go somewhere where it's easier to sample. You would, and potentially less turbid as well, so that you could actually use those photoquadrats or subtidal photographs. And I would agree. I would agree with selecting validation sites quite carefully as well.
Question about the turbidity. You showed us a picture of murky water. The waters aren't all that murky all the time. I mean, it's a lot murky a day or 2 or 3 after a rainfall. Sometimes you have a long dry spell, the water is much less murky. And a GoPro can get close up. I was involved advising a project last summer in the East River where they use the GoPro, and you get close to the substrate to the bottom, and it's pretty clear. We didn't go out.
You got really lucky.
Yeah, for our particular,
You get very
yeah,
close. You have
you
a
do.
long pole, and you get it down,
And we had our students design that. It was very exciting for us
in
all.
the East River.
So,
For our particular purposes, the photo quadrats using a GoPro weren't practical. There were, for a number of reasons. not just the turbidity, sometimes it was accessibility, sometimes it was analysing the actual digital data that we were getting out of it. We weren't able to do it at as fine a scale as we could on the settlement tiles. And so we steered away from that, although we did invest quite a lot of time, quite a lot of resources in testing that out. As you can see, we have our interns there with our constructed device to house the GoPro and also to take lots of water samples. So we understand that it's not that turbid all the time. We also understand for our particular purposes it wasn't a suitable method.
I just want to go back to the statistics that you saw differences among sites. I think you said it was total community. So is that, is that everything that you sampled? And did you try to break that down at all and like just look at like benthic invertebrates and see if
we missed out on being able to do some really robust stats on this because we had so much replicate loss, and we acknowledged that. And so we couldn't do, for example, a permutational multivariate analysis of variance because we didn't have enough replicates. Of differences in community, but we weren't able to follow that up with some more robust analyses, and that's definitely something we want to do.
Christina had a question and she's been waiting.
Yeah, Christine.
What are you trying to measure in terms of rocky intertidal habitat? I mean, in New England, there's a lot of rock. Here, are you trying to mimic oyster reef? I was reading a paper in Frontiers in Ecology and Evolution recently by Horn, and I was really struck by their statement that marine urbanization is changing the flora of urban estuaries by encouraging species that grow on herb substrate. And they were discussing the idea that if engineers and ecologists were to join together, they could work to create better protective structures that could enhance what we're losing, rather than invite changes, including introduction of invasive species. So, where should we be?
Okay, so the question from Christina was, I guess, are we trying to measure hard substrata, and are we trying to kind of, we're trying to mimic hard substrata, or are we trying to promote hard substrata in a formally substratum environment? Do you mean of the protocols or of the shoreline development? Because we're not in the business of shoreline development.
If you assess the community that lives on any of these hard structures, what is success?
Hang on.
I have a little bit of conversation.
So
success for us is developing a protocol that represents, I understand, I just want to be clear.
Yeah.
So success for us is developing a protocol that mimics the surface we're on so that we can measure these things.
Yeah.
But success for the harbor
harbor
restoration
is
a whole other ball of
question.
wax, right?
Yeah,
Kate, speak a little to that. So it's a very important question. I don't think we necessarily have a specific answer yet. The purpose of this study is to start sort of building a library of case studies so that we can get at that question. We know by a number of other research that has been done that things like wetlands and sort of more shallow slopes are better ideas no matter what. But given the urban condition and given a space like Battery Park, what can we do? I think this is sort of an ancillary question, but what can we do in those areas, and how do we measure those differences? So I think the sort of short answer is we don't have an answer to that yet, but that this protocol is a means to start cataloging different case studies, and we say, okay, it's representative. So what is success within this? Which may seem a little bit reverse, but I think we don't have a way to understand yet.
I guess in the absence of a way to reconvert all hardened shoreline to soft shoreline, and the fact that we are adding hard substrate into a formally soft substratum dominated community, we're creating these areas where potentially invasive species, and by the way, most of the species we found in the sessile plates were non-native species. So yes, we are doing that, but we were not specifically in the business of figuring out what is the better shoreline. We were figuring out how do we measure the communities on the actual shorelines. But I know it's an important question. It goes beyond this particular discussion.
The impetus. Yeah. So the impetus for this project, more diversity. There's probably other indicator ways that we could measure success, and that's something that this project started to do. Is have this fish food index, have an index so that we can kind of try to compare.
Too much to talk about.
But I think the thing is that there's a lot of people proposing alternatives to existing seawall shoreline types. In fact, we have somebody in this room that was involved in the design of Harlem River Park, which is supposed to be sort of an ecologically sensitive design. But we don't have a way to measure that going forward. And
This question is in no way a criticism.
oh no.
Oh no. It's just like I think it goes beyond
Yeah.
today.
The next question.
It's the next one. Yeah, exactly. I mean, yeah, we're introducing hard surfaces.
Right, and how can you make that as ecologically beneficial as
That's right. But you're right. It's an interesting question because like some of these shoreline modifications that are going on down in Staten Island are creating hard shoreline where maybe there wasn't hard shoreline.
And it may really be
What does that actually do to the full community, and what does it do to the system?
It may really be that the outcome is that it's not as much about the surface as it is about the shape. That we have.
That's true. I mean, we have
if want
shape
to
considerations
create-
too.
If we want to create a more diverse marine environment, that we actually have to reshape the shoreline to mimic some of the natural shapes, not simply resurface it to create more fine scale.
And then more value. It goes beyond today, Christina, and we acknowledge that. It is.
It's a measure, but it can be a simplistically applied measure of success, and I think maybe a more suitable type of idea would be to try to look at the diversity which was lost, the specific diversity which was lost. In some places, more isn't more. Some places, lots of biodiversity with
or differences in functioning, ecosystem functioning rather than like straight out taxonomic richness. There's a huge suite of questions there about exactly lost when. What baseline are we talking about?
I think that's
we're getting into the.
I think that's harder to get. I think that's one reason why we're leaning towards an index, and we're just very excited about your question, which is why we're responding so much. I think it's very very important. Yeah.
Phil's question as well is that Phil Parker and his colleague Shalon Politon are architects who teach at Pratt, and they approached Stephen Handel about whom I work with at Rutgers about and Arup engineering firm about creating better hard structure habitat, both ecologically and for social concerns, to be a better experience for the public, and to be a safe structural environment. So, we're gathering all of this information together, and we're saying, what does that look like?
Yeah.
So that is fueling
it's
these questions.
This is an exciting thing to be involved in. There's so many different groups that can contribute.
But integral to that is exactly what you're doing, is the testing, because we can make all sorts of assertions without any.
Without knowing if it does anything.
And an architect is very good at that.
That's right.
We're not bad at this.
Yeah, just super quickly, the fish food index was just one of a range of different indices we were trying to use in order to measure sort of overall kind of habitat quality. So we would measure like two functional indices as well. So we had filtration as a core function of some of these communities, and we had bioconstruction as a core function of hard substrate communities, as well as fish food suitability. So it was something that we're building out with Dave Strayer and a couple of other undergrad student volunteers from Columbia, and it was something that we started for this protocol development, but something that we can keep talking about as an alternate or complementary measure of habitat quality.
So maybe let's take a couple more questions and then we'll close.
So I have a question slash comment, I guess. So to summarize the use of this protocol, assume validation occurs and this is a perfect application or tool to gather research. This is something, this protocol would be used by Hudson River Foundation or other advocacy groups to do research and gather information on the best type of shoreline treatments that then would be used to, discuss or promote to the city for installation. Is that right?
I think
So maybe
think that would
you can
be-
answer that, but maybe
Is that
we need
like
to repeat the
like
question
this?
again.
If you're monitoring, if you're implementing the protocol like Harlem River, which was an ecologically forward-thinking shoreline, if you're monitoring that in the future to determine if that solution was a viable solution, if it makes sense to implement elsewhere, or maybe if the gabion baskets are better than the concrete riprap baskets, then you can advocate for more gabions in locations where that makes sense to install over a standard riprap.
Yeah.
So is that where this is going?
That's-
Because I
So Brian was
can't see
asking,
where we would use this
what's the
Sure.
end goal of a
elsewhere.
protocol?
Like I'm
So-
not gonna,
Right, so
that makes sense.
my answer would be, so Brian's asking, where are we going with this? What are we doing this for? Assuming that the validation works out, is the point of this to use this to decide what is the best surface for every time we rebuild, when we rebuild something? And my answer would, and I'm just me, but my answer would be yes, but that's an application. I mean, as a marine scientist, I would use this to simply understand I work up in Piermont. We don't know what the community of sessile invertebrates is up in the Piermont area, and I would use this to understand that and compare Piermont with Cold Spring, with Manhattan, with Sandy Hook, and just simply to understand the diversity of life at that level. I mean, so from
I think just to build on that as another application, going back to what Marcia was saying, is I think you could use this to establish those kind of baseline quality conditions across the harbor. And so you could identify reaches of the harbor or areas where some sort of intervention, whatever that intervention might be, would be more or less valuable. I mean, maybe that there's certain parts of the harbor we could, design flower pots on bulkheads up the wazoo, and it's not going to make a difference because of other things. And so I think there's that. To me, that would be another application of this beyond the baseline.
And just as a add-on to that is, so let's say just for example, the gabion baskets turns out to be the best solution we've implemented yet, and Hudson River Foundation says, hey EDC, every time when you have a chance, is more fill than another solution. So then in my scenario, I'm serious. When I go to DEC and Army Corps, I have to argue why I'm putting in a certain shoreline treatment. And if that shoreline treatment is more fill or traditionally more volume or something like that. So then we could use this research as backing to say we're implementing this because of the work that came out of Hudson River Foundation. And to that, I would ask Susan if it's part of this is that it's non-specialist. So if the research is done and a non-specialist put together the reports that then we are providing to DEC and Army Corps as our foundation to implement this, is that still viable from a DEC point of view? Or do they have an issue with that? Or should it only be done with specialists? I mean,
Always do the testing with specialists.
I mean, okay.
I don't know, I'm just
So
throwing that out there.
So my experience with with this is in the realm of nutrients, right? So I work with plenty of citizen scientists who do nutrient measurements in the harbor. I also work in the harbor in upstate. I also work with the DEC scientists. And, when the DEC has a specific question they want to answer, they send their own people out to do, or they hire a lab to go out and do measurements. They're not going to give the same weight to my high school teachers.
Why not create a protocol that the DEC agrees with? So they
Yeah,
don't.
this protocol would be the same no matter
Right.
who's
Yeah.
using it.
I think,
And this would be an additional validation step, I guess, in that you could, and we did quality control, QA/QC, and the results.
yeah, I think, Brian,
We got a 2% error rate for our cross validation of our actual identifications between what the students were doing and what the experts were doing.
Specialists. So between non-specialists and specialist groups, it's about a 98% agreement in terms of our IDs. So we were quite confident that our protocols were robust enough in order to be utilised. So the results from our protocols would be similar across non-specialist and specialist groups. But of course, if we could do further quality control and validation on that, then that would increase the confidence of state departments.
See what I mean? Like,
No, but it sounds a bit hypothetical to me. If the DEC has a sessile invertebrate shoreline monitoring protocol that works excellently and has been validated, but requires a level of expertise that we've engineered out of this, then that's a practical question we could discuss.
Right.
But I'm not aware of the DEC having such a protocol.
Yeah, I think just to step back, again, the purpose of creating this protocol was multifold, but we had sort of a couple main impeti, and then there are sort of all these other ancillary questions that are interesting from a marine biology and other point of view. So number one, if the more information that we can add to this, so there doesn't currently exist a way to measure these types of sites. And so if we create that, then we can add a layer of information so we all as a community understand. I mean, that's one reason why we've engaged DEC and DEP and others, so that we're all sort of providing that input into this protocol. And I think over time we would like to, yes, understand it from sort of the Hudson River Foundation and science view, what our different options are, which ones are better, but also as people propose new types of construction and alternatives such as the Harlem River Park, that DEC can say, okay, we have this protocol that was developed in addition to these things. When you're implementing it, it will allow you to do it, but you need to monitor it for 3 years and contribute to our shared database of different shoreline types.
Maybe we created a little confusion by talking a lot about these high school kids. We didn't design a protocol for high school kids. Our target audience, and we said, who's going to do this? We were thinking about an EPA or a DEC or a USGS
Management
field
groups.
office, where you have field techs and, professionals, but who are not necessarily marine ecologists, specialists, or they, not necessarily, so, but that was our design spec. We didn't dumb this down, for middle school kids or something.
Yeah,
Or,
this
we
provides
designed this
ecologically meaningful
for
data,
field
like
techs,
robust data.
but who don't happen to, and we were then ecstatic to find out that, teachers and high school students, could play a role in this.
I think one of the most troubling things about it is that you could legitimately, I think, take from your data that there's no advantage to doing a replication of Harlem West Park based on.
I'd be cautious. As I said, I'm cautious with the level of replication that we lost. We can't make solid conclusions. This was very brief in the report. Look in the report.
Right.
We can't draw all the conclusions that we would like to out of this, but if it becomes a public piece of information,
Which it will.
in some form will, you could expect that people like our bosses, not technical marine ecologists, could say, why should we do another Appian wall? Let's just do the same
My-
well, it makes no difference. There are other aspects, porosity, flood storage, other, algae. But as far as just like habitat for marine animals, it's the topsoil. Why bother?
May I stress though that the testing we were doing was to develop the protocol, not to test differences between the shoreline types, and our results need to be taken with caution and with consideration that we lost replicates, but should never be used to advocate for certain shoreline types
I
to
mean,
not to.
Right, when we, wait, no, I understand, but when we say, right, so maybe you don't want to publish this. But
Okay,
when we
yeah, we
say,
could
when
pull
we
out the
say
results
this
if you like
lacks
to, but
statistical significance, we're not joking. That's a real statement.
So
What?
I think
I'm not publishing it. We will not publish it as an analysis of different shoreline types. And if someone was going to pull out something from that and advocate.
By so many in the public realm, that's an irrelevant point. It's just,
Right.
that's a detail
Yeah.
that goes way beyond.
I think
If you want to study, okay, what we've done, okay, right, I understand, but I'm saying this is a protocol development agreement, and we've developed a protocol. Now, if you want to do a study, you have to actually finance a study and do a study. And that's just true.
So for those on the phone or on the connecting remotely, I think Marta Johnson asked about concerns about publishing the results of this project. Simple conclusions based on the data results that came out of this project. I think we've talked about this before. The focus of this project is really to develop the protocol, and the next phase is where we would get into, well, can we use this to answer that question? We absolutely want to answer that question. We can't, given the data that we have. So I think we just need to be clear about it when we publish it in a public way, which I think we should. But I think we just clear that we're caveating it, and that these results are differences between areas, but we can't say that there are differences between actual shoreline treatments. And that became very clear out of the results of the project. So I think we just need to be very clear about that.
Just make a suggestion of when you publish it, and you usually have this conclusion section or like in a nutshell section or something.
Executive,
and maybe
yeah.
that point isn't one to emphasize. You talk about developing the protocol, you talk about the different people who can use it, and then it gave you big differences it showed that there was variability among sites, and maybe you don't say anything about, whether or not you found, results between.
I mean, if I were to publish this, I would simply and purely publish it as a protocol. And my point about the sites would be simply that the protocol is sensitive enough to distinguish between sites. But I wouldn't even publish it as something that states unequivocally, this site in the New York Harbor has more diversity than that site in the New York Harbor. We don't have sufficient data. I mean, you haven't, as Lisa pointed out, if you look, you have the 4-week sample, the 8-week sample, you have significantly different communities. These are and all of us who've looked at ecological data know that you have an enormous amount of temporal variability. We don't know what we have yet. You just can't say that.
And maybe since it hasn't been validated,
Yeah.
you really can't say that.
I think we can just be very honest about it and maybe even actually be upfront and say, we're not yet there about sort of being able to compare different treatments.
Yeah. Is that the next study?
That's what we hope. I think we need to sort of reconvene internally and take input from you. I think this is a good place to wrap up. Thank you all for all of your patience and excitement about this topic. I think I see people's sounds elevating as they get more excited, which is really great. It just shows how much interest there is in this topic. And I would encourage all of those online and in the room to, as you think about it, send some more questions. I'll try to get them answered. And thank you for Elisa and Bob and everybody for presenting today.
Thanks, everybody.
Thanks.
Yeah, yes, we have not posted it online yet, but I think I'm going to go back and actually look at that question of caveating it, and make sure that that's clear.
Yeah,
and
we have
then we'll-
a report and a protocol manual and a video as well. And some follow-up additional water quality measures to be produced as an addendum once the lab gets going again. Yep. Happy to follow up.
Thank you all. Oh, yeah. Can we go to this
Recording.
on Melissa?